Again, thank you. Actually, I'm going to do a little slightly different than the title because in the context of the last two talks, both speakers spoke about the safety, risks, and benefits of stem cells. So a part that was not discussed is we have all these new concepts and lots of new CAR T cells and other therapies. I'm going to focus a little bit what happens behind the scenes. You know, the labs or the facilities that prepare these CAR T cells. The area that has actually lagged behind is the engineering, the hybrid of biology, medicine, and engineering to create these cellular therapy products. So I'm going to try to focus on how to marry these two to create cellular therapy products, both in the context of blood cell therapies, because most of us are talking a lot about blood cells because these therapies have been done for 30, 35, 40 years. So we have a lot of experience. But then towards the end of my talk, I'm also going to spend time beyond the blood cells, especially at my institution where we are trying to develop other cell therapies. And I'm going to show you some other things. And these are not normal. These are very unique protocols and procedures, a few places in the world that's been done, but where we can go. In this context, one of the programs we have is to try to come up with alternative therapies for diabetic patients. There is 10, 15 million patients with diabetic in the US. So this is like a beginning, how to try to replace insulin by giving islet cells, cell therapies. So I'm going to focus on those two aspects. Before getting into that, many other stem cell therapies, actually, the biggest worry, especially using embryonic stem cells about 5, 10 years ago, was teratoma formation. You give patients these cells that are so primitive stem cells. How do you make sure they don't get other cancers, teratoma formation, tumor formation? So I think still those are concerns, but many of those concerns have been kind of addressed in terms of safety. But the other aspect of the safety is what Dr. Bambusin alluded to and Dr. Rodriguez, like you give stem cells. For these stem cells to be taken into the body, you need to condition these patients, and not engrafting the cells is a big issue. So those are some of the safety concerns that people with drugs and other means trying to address. But then coming back into this arena where we are trying to create these cellular therapy products, and this is where, again, regulations become very useful, is how do we streamline to get processes in place to give these therapies. So that's what I'm going to actually focus on, give two kind of environments these cells can be prepared. One's called GTP, that's called Good Tissue Practices Guidelines. These have been developed over last 20, 30 years. Most of these practices are in place for producing small molecule drugs, like the Lipitor or any other drug you take is very well regulated from raw materials all the way to be getting into a pill. Similar principles have been applied for cellular therapies, although it's very challenging because you're talking not an organic molecule, you're talking a live product. Live cell, you have to keep the viability and the sterility and the potency. So these are the issues that we address in the background in a lab or in a facility. And so I'll give you some background of in our facility, how we do those things. Some are very simple concepts, actually. The context these become important are because it's different when you do 10 transplants, 10 therapies, 15 therapies. So for example, at my hospital, we do between 200 and 230 transplants. The lab that I run see 600 cellular therapy products go through my lab. So when it becomes that type of a production facilities, minor things become major. So like we have 2,000 stem cell products cryopreserved in my lab, all the way going to 1987. So how do you make sure you don't mix up these samples giving the patient? the error, the room for error is almost, zero essentially. So those are the issues that we address as we go through. And then also I'll give you some of the aspects, actually build on Dr. Wambusin's talk, T-cell manipulations. Some of the slides will be same. You could tell we worked together in the past life. So now we are two different institutions. And then the last part, I would talk about this islet cell isolation, which requires cGMP environment, which is called good manufacturing practices environment, which is essentially like an OR environment. And so the functions, goals of a clinical cell processing lab. Actually, the word lab, I'm trying to change the rest of the world not to use a lab, because when most physicians think of a lab, it's a place where you test things. This is not a testing place. This is actually a manufacturing facility. Actually, the FDA regulations are written as a manufacturing facility as opposed to a testing place, lab. But the functions of a cellular therapy production facility is multifaceted. So we manufacture, which is like a pharmaceutical company. We store, like a warehouse. We dispense and infuse, like a pharmacy. All those functions are done by the same group of people, same staff, same mechanisms. So how do you regulate these things? And since 2005 in the United States, all facilities that do these functions have to be registered with the FDA. So it's just a registration. We just tell them what we do, and that gives them permission to show up at our facility unannounced. So for example, I've had 2 visits. They come and knock on our door. They don't tell they're coming. When they come, they don't leave for 5-6 days. So they've looked at 600 different patient charts in my lab last time they came. They look at everything. So that's how those regulations in the United States work. They are the only entity that have the power. When they leave, they can shut you down that day. Usually what they do is, I'll talk more tomorrow, is they will give you some observations that they had. You have a couple of weeks to reply. And if they like your responses, it's fine. And those things are public. You can go to the FDA website and look at every facility in the United States that are registered, what their observations were. So it's public information. So it's very transparent. So anyway, so a facility like that, this is for my particular facility that I run, but it's pretty common to most other academic cell therapy or hospital facilities that produces cell therapy. So the middle is a lab. So if you don't think about this, the cellular therapy product shows up at the patient's room like Dr. Rodriguez has shown. You hang it. It's a very, very, not like an organ transplant. You hang it and it goes into the patient. This is everything that happens in the background, actually. Of course, cryopreservation is very big. Thaw infusion is very big. So these are very critical steps in the process, but all the others are also equally important. Transportation, I mean, we get cells from all around the world because my facility, the transplants we do, we have a hard time finding matching donors. We have a lot of Asian, African American, Hispanic patients, so we search all over the world. So sometimes they come from all parts of the world, and their lifespan is very, very short. So we really pay attention to all these factors and functions. So, I mean, this is actually my staff that works in the lab. And all of us together have over 60 years of experience in doing what we do. And when we hire somebody, you don't go to school to learn this. So actually, they don't touch a patient's cells 9 months into their employment with us. That's how much we train them, because there's no room for error. So again, these are the basic fundamentals of what we do in the facility. This is what FDA regulation, and having an accredited, regulated facility is so critical because of these are the parameters we pay attention. Sterility, again, it's a live product. You cannot autoclave them. You kill the cells. Identity is important because this is more and more becoming important. Most of our transplants that we do, it's not a pure population. It's all different. CD34, which is a stem cell, it has T cells and other cells. So identity is becoming more and more important in defining what we give. Purity is also very important, how pure is. So the Dr. Wambusin just alluded to CAR T cells. So this is such a breakthrough finding, but the cellular therapy product in a CAR T cells, after modifying the T cells, really is only 20% are real drug actually. The transduction efficiency to get the genes into these cells are so difficult. So it's not purified. Even the one that the FDA has approved, only 20% of that cells is a true CAR T cell. Rest are passenger cells that have never been transduced. So that goes into this purity. Then potency. In other words, how well do these perform? How potent is the dose? And then the stability, how long can you keep those? So these are all the parameters that we pay attention to. And then, but again, like I mentioned, the challenges are so different from a small molecule drug. And it's our lot of each, I mean, if you get, for example, Lipitor, each lot might be 100,000 pills. Our lot is one. Most of these cellular therapy products made for one patient and one patient only, including the cord blood cells we get for transplant from cord blood banks around the world. For some reason, if we don't use it on these patients, we throw it out. We cannot give it to somebody else. And we buy these cord bloods. Right now, a cord blood product is close to $40,000 US. So that's the other aspect of things that we don't discuss. Even CAR T cells, a different forum, but these are all the things that we start thinking about. I mean, we have such cool findings, but the newest approved CAR T cell product from Novartis, the cost is $450,000 US. That's how much they're charging. So, and that's before all the side effects. You have to treat these patients for 3 weeks. So they all go into this mix of deciding how to bring cell therapies to people, the cost, efficiency. So you're talking a patient with who get CAR T cells, usually it's over $1 million US, 2 to 3 weeks in hospitalization and intensive care unit. So anyway, so these are all the aspects that we pay attention to. And so this is our standard, how we collect these cells. And so after cells are collected, this is where most of the things happen behind the scenes, graft engineering, and also regulating the number of T cells in this graft. This I'm talking in terms of now blood cells. And so I just want to go through some of the things we do to this product before it gets into the patient. So we call it like a life of a stem cell product. So these are the raw materials. It can be bone marrow, cord blood. Mud is the match unrelated donor product that we get from around the world when we don't have a donor. So they all have different challenges. So these are all the raw materials. And typical easiest thing we do is to volume reduce, cryopreserve, or give it as a fresh product into the patient. But all these testing has to be done. CD34 cell marker is still the gold standard for how many stem cells are in a product. And then viability, microbiology testing, HLA testing, T cell enumeration. So our facility does a lot of manipulation, not just standard. These are some of the things we do. This is all done through a physician's prescription. So the transplant physician prescribes what we should be doing, and then we prepare them and also select for different cell populations. So just to give you an idea, and everything is done in a closed system. So these are class 100 biological safety cabinet. It's even almost cleaner than an OR room. Everything is done through this coupling system. So none of the cells see the open air. It's all done through tubing systems and bag systems. And this is an instrument that came about now, I don't know, 15 years ago. Actually, using this instrument, it's called the Clinimax unit, you can purify the CD34. So this alludes to one of the factors I mentioned is purity. So rather than giving cellular therapy products that have 3 to 5 million CD34s per kilogram weight with lots of T cells, now we can purify to 95% CD34 cells. And now we have more control of the patient cell populations to give it to the patient. And also, this has become actually one of our flagship protocols where we give a CD34 purified from this machine along with the cord blood unit. It's called HaploCord, developed by Dr. Vambazine. And we've now transplanted 189 units of cord blood along with the CD34 in our facility. And The nice thing about this is you take care of two issues. So the cord blood is the one that engrafts long term. Short term, we give a haplo-matched haplo product, which does the bridge. But also what we can do is to add back T cells. In other words, we are customizing the cellular therapy product for these patients. And then the next challenge is for this CAR T and other cell therapies to fight different leukemias and cancers is how do you grow so much T cells? How do you grow even stem cells? How do you expand them in the lab to get a cell dose for these patients? And especially now that the CAR T cells are really becoming more and more useful, and so how do you grow them? So we need engineering mechanism to grow them without losing its viability, without losing its potency. So, and this is just to show how you get T cells from a normal person. Our T cells are not activated. So some of the ways to do is you harvest these, and in the lab, in a facility, you can expand them by exposing them to stimulated like CD28, interleukin 2. So those are some of the things that we do. So for doing that, we need platforms. So there are a lot of whole group of people who are developing these platforms. So I want to share some of these platforms. So for example, Novartis product that's been now become standard of therapy 2 weeks ago, they use this platform. It's called Wave Bioreactor, where you can get billions of T cells. So we collect our patients' T cells without giving any growth factor, ship to Novartis. Actually, in the Novartis case, we cryopreserve. It's a cryoed cellular therapy product that goes to the company. They expand them. They thaw it and expand them here, and then send it back to us. But also there are other ways of doing it, and one of the ways of doing it is these types of gas permeable bags where you can grow and expand the T cells. But again, while we do that, we keep in mind the the sterility, viability, and potency. And one thing that we really, these are the three platforms that many places are using. Usually the most complicated thing is not the best. So we really like this really simple system called G-Rex bottle system. It's a very, very simple but very innovative platform. So one of the biggest bottlenecks for cells to grow and expand is ability to exchange air and nutrients. So this company, they came up with this idea where they created this bottle, but at the bottom you can create a huge media volume, still the nutrients go through, but how does the air get through? Actually, they put a silicone gasket at the bottom, so all the air comes through the bottom. It's really good for blood cell growing, so you can get billions of cells within 10 days. So this is how you can get them to grow, and it's also in a closed system, although it's not completely automated right now, so that's kind of a bottleneck, but otherwise it really works really well, and it's inexpensive. So the reason also I want to share this is more and more sophisticated therapies we build is cost is becoming a big factor that it cannot get to all the patients. So creating the devices that are relatively cheap is also as important as coming up with new concepts. Otherwise, it's of no value if a therapy costs $1 million for one treatment, it becomes a big issue. So anyway, so this is just comparing different platforms and showing how you can get millions of, this is over 1.4 billion cells within 10 days. So in other words, the that Dr. Van Bussing alluded to, it doesn't take 2 to 3 weeks. Instead, you can get the product back to the patient within 10 days. And this is a system that we're going to adopt pretty soon. We are in the process of building a new facility. And this has all the good features of all the systems, actually. It's a bioreactor. So essentially allowing automated cell seeding, feeding, and harvesting. Also has everything is in a closed system and automated. And you can actually expand. The biggest use for this is actually mesenchymal stem cell expansion. For a lot of the therapies I talked about this morning, this system is being used right now to get lots and cryopreserve whole banks of cells. And again, these are clinical platforms. So using these kinds of devices that already FDA has vetted. So there's a whole separate stream of regulations how to get approval for devices, which is different from drugs. And each of these together, working together, will allow you to come up with a really good cellular therapy products. And this allows you to get 45 billion cells from a single run, which gives enough cells for lots of patients, especially in the context of MSCs. Anyway, so I'm going to now switch gears from blood cells to a little bit of regulations, and also how do we expand our experience of blood cells to other cell types. So the example I'm going to talk about is islet isolation. This is exactly not regenerative medicine. Some ways it is, some ways it's not. So it's not like starting with a stem cell, which would be the ideal. This is actually isolating islet when an organ is taken out and transplanting into a patient to cure diabetic patients. So this has been one of the more complicated and more messy processes we do in our CGMP facility. So everything I talked to you so far is not done in a CGMP environment. The environment is not like class 100, not an OR. This has to be done in a very clean room. So that also actually increases the cost, but it's really critical. And again, so this is a good example where how long it takes for processes like this to develop. So we've done this for 10 years. Now we are 1 year away from applying to get what's called BLA, Biological License Application. Once you have a BLA, now it's an approved commercial product, just like a approved small molecule pill. And in order to get to this place, we have done over 100 islet isolations, and every isolation, every piece of data is reviewed by the FDA. And you have to provide your process, how you've done it. You have to give the outcomes and the clinical outcomes of these patients, follow-up data, and the environment that these were done. So the environment this is done is this facility. It's been functional for now 13 years. And it's where people gown up right here. And then the materials go through a separate area. So the islets are done in a special suite right here. All the other cell therapies are done in this part. Islet isolation is done here, where personnel move in, and the isolation. So there are two types of islet isolations we do. One is allogenic islet transplant, the other is autologous islet transplant. Actually, autologous islet transplant is standard of therapy. So you can actually get reimbursed from insurance companies. And what kind of a situation do we do islet transplant under autologous setting? When patients have pancreatitis, surgeons take out the pancreas. Those patients' pancreas come to our lab. We isolate the islets. It's an 8 to 10 hour process. And like I alluded to, islets are clusters of cells, so you cannot cryopreserve them. So after 8 to 10 hours of isolation, it immediately goes back to the patient and gets infused. And just like bone marrow transplant, unlike an organ transplant, it's very anticlimactic, the islet transplantation. You just, it goes through a portal vein, through an IV, and it goes and sits on top of the liver of the patient and starts making insulin. So, but the allo transplant is a lot more different. Logistics are a lot more challenging. Somebody else has to die. So auto accidents, surgeons fly and harvest the islets out in the field. I mean, harvest the pancreas and brings it to our facility. And people are on call, and then you do the isolation. So this is inside the facility of the isolation process. And these are the staff that works there. So it's completely, you have to gown up. And the benefits of this program is, especially auto transplants, are really valuable. So Otherwise, pancreatitis patients, I'm told, becomes diabetic, even though they were not diabetic before pancreatitis, after the pancreas is taken out. So this prevents them becoming diabetic. And then, so we are at the verge of this BLA submission. We've undergone the phase 3 trial. This is also very unusual. Usually these things are done by pharmaceutical companies. We've done this as an academic facility. So if we do get the BLA, we would be the first place in the United States who has a commercial BLA license to do islet transplants. Because of the cost and the amount of effort, many places have actually stopped doing that. This is still not the final cure. I mean, this is transient, because still, like any good thing, there are still drawbacks. So the biggest drawback in this therapy is after 2 to 3 years, I think about 30 to 40% of the patients lose their islets. Your immune system comes and clears it, but some patients don't. The person who's taking the lead role in this is Dr. Witkowski, who was trained at Columbia and come and set it up about 8 years ago. And his team does all the, he's the surgeon, and his team is fully trained to do this, and they come and work on this place. So the process is you get the donor pancreas, isolate the islets, and then put it back and through the portal vein of the recipient. And just a few slides, and then I'm going to finish up with a 2-minute, I know it's before lunch, but it's still important to see how from beginning to the end, it's a 8 to 10 hour process in 2 minutes. From the time we get the whole pancreas, from an entire organ, how do you get cells? So in order for all these 10 hours of work, we have to have 300,000 islets if we are to put it back in a patient. So that's our cutoff line, 300,000 viable. We do a testing to see whether they produce insulin. If they do, we take it upstairs to the patient's room and give the, so it's a long process, and there are lots of failures, but there are lots of successes too. And in a nutshell, so at the end, this is what the islets will look like. But it's a lot of digestions and centrifugations. So there's a whole industry, so everything that this organ touches is clinical grade. All the enzymes, all the instruments. I mean, most of these were developed not by companies, by the surgeons themselves. So this is completely homemade. So original idea came from Canada, Edmonton, Canada, who originally did this procedure, but then it's been improved over the last 10 years. So everything is pretty much a homemade process and lots of practice. And this is how the people have to dress up to do this, and everything is done in here, all through tubing. And it costs us about $15,000 US to isolate one of these islets, which is not bad considering how much effort it's required. And then, so this is the units that we need, the final testing, usually 300,000 islets, 50% success rate in most experienced centers. It's risen to a little more than that now. And then this is showing how it's ultimately infused into the patient. And then all these tests are required by the FDA before we give it back to the patient. And then these are the final islets. And this is Dr. Witkowski doing the transplant at the end into the patients and through the portal vein, and using guidance by IR guidance. And this is like 2.0. This is the vision going forward. Like I mentioned, large majority of the patients after a few years get rid of the islets. The newest way we are trying to treat these patients is, along with the islets, we also isolate their T cells. And there are a subset of T cells in your body called T regulatory cells, which expresses CD25. If you can purify these cells and give it with the islet, now it keeps your immune system under control. So this prevents these patients getting typical immunosuppressive drugs, which are not good long term. So the plan is to, the vision is to create islets with tregs, or get the islet cells and coat it with T regulatory cells so the immune system doesn't recognize them, or put it in a pouch, a surgical pouch, and implant them. So this is still about a few years away. that we are trying to develop. But overall, islet transplant itself is, right, when it works, it truly works. And then I don't want to bore you with all the numbers. Lots of numbers go behind what I showed you. And especially for Treg isolation, we use that same Clinimac machine in my lab to do the debulking. Then we do flow cytometry sorting. We sort for a day and a half in the cGMP to get the enough T cells, and they are expanded into Tregs, and then cryopreserved, ready to be given along with the islets. And then this is a patient that got 5 insulin, and she's been off for 5 years, and she can now eat cupcakes. Anyway, so, and these are the other nurses in the team, and this is the islet transplant team who works in the lab to isolate the islets for 8 to 10 hours. And it's the PI, principal investigator of the project, Dr. Witkowski. So I'm going to finish it right there and show you the, I hope it works, the A to Z, the 1.5 minute movie. So unlike the blood cells, this has to be done in the open, but it's under class 100 sterile conditions it's done because the initial steps are pretty messy because you got to, the other issue is it's so critical the surgeon harvesting this organ in the right way. If you don't have all the veins and the arteries intact, we cannot do any of the perfusions of all the enzymes to break down the tissue. So usually if the organ is harvested in the right way, you can inject all the enzymes and it becomes about 10 to 15-fold bigger, and then it digests over time. And then it also requires coordination of at least 3 to 5 people at the same time, and a lot of quality control at each step. Those devices he's shaking, they're all home developed. They are not, you can buy it from anybody. And then always there's somebody who checks the quality of the islets at every step once you get to the point of the islets. And you'll see so for separate, this is so that this is the final islets that goes into the patient. So I'll stop right there. Thank you.